I mentioned briefly the Boundless Expedition, 1803 to 1806, which concurrent with the Lewis and Clark expedition, although it had nothing to do with it. It was an attempt by Charles IV to vaccinate everyone in the Spanish Empire. And he took 20 orphans, and our 22 orphans, put them on a couple of ships, and they sailed around the world. They would infect one kid and then use the pustules from that kid to actually vaccinate. It's a fascinating story, and I think the best introduction to this is actually Julia Alvarez's beautiful book, Saving the World. She's a wonderful Latina writer, and this is a very moving book. Well, everyone's talking about vaccines now, and traditionally it takes anywhere from 5 to 15 years to develop a vaccine, and it's a very long and involved process. It is being expedited as much as possible, and it's strange since we had started a vaccine for the Sars, but then stopped it. We are in phase 3 of a few clinical trials. So phase 3, which is actually human testing. And then later, hopefully this winter, we'll get into a series of complicated reviews and then manufacturing. The goal is next summer. We'll see if that happens. But anyway, that's the goal. And where are we now? There are over 141 vaccines in development around the world, China, also in England and elsewhere, India. There are only about 9 in phase 3, and 1 has been stopped. 1 has been technically approved, but then not in the United States. So that's a little kind of mute. So we should see something probably in next summer, probably not before voting day. Plus, after licensure, there comes a whole series of hurdles. You don't have to worry about these, but the CDC and FDA and also National Institutes of Health and so on have to sign off on all of this. And so there's safety and all sorts of other things that have to be done. So this is as involved, and perhaps more so, because it also is looking and analyzing all the projects up to that time. It is really fascinating now to look at polls, especially if they're broken down by Democrats versus Republicans. I don't know how we have to divide everybody this way, but anyway, according to this recent poll, 40% of Republicans would not get the vaccine. I don't know what that means, but maybe they must believe all their own propaganda. If you look at this, it's estimated that anything from 30 to 40% of the population would not get vaccinated, which is self-defeating, because then probably herd immunity would not be achievable. And also, we know from all the fake news that Bill Gates wants to put microchips in the vaccine, and that way they can track everybody. They can track everybody with internet searches, but I don't know where this particular bit of wisdom came from, but there's so much nonsense out there. The R-naught. The R-naught is simply an estimate of how many people will get sick from contacting another sick person. And to give you an idea, the influenza that we normally get every winter is estimated at 1.3. COVID is anything from 2 to 3. So it is at least minimally 2 to 3 times as effective. But then when you compare it to something like measles, measles runs anything from 11 to 18, which is incredibly high. Even Zika is much, much higher. So the R-naught, and it's an average. I recently read about a case of a party, and one person infected 60 other people. So no masks at a party, and it is very likely to have some serious consequences. Now, the R naught is used to figure out what is called the herd immunity, which is the minimum proportion of the population that needs to be immunized to obtain a restriction to how the virus can spread through the population. There is a simple formula that's shown up here, and it refers to the fact that either everyone has been sick and has recovered, or the population has been vaccinated. And using the fact that it is R naught of 3 for COVID, then about 70% of the population, meaning more than 200 million people, either have to be vaccinated or have recovered from COVID to prevent the virus from actually getting through the population. Doesn't mean everyone is immune. It just means that the virus can't find entrance to get to these people who are vulnerable. And it also depends an awful lot on how efficacious the vaccine is and what the rate of vaccination is. So if the vaccine efficacy is low and very few people get vaccinated, you're not going to end up with herd immunity. If the efficacy is low, but the vaccination rate is high, which is very likely what's going to happen to us, we don't know, there's still no herd immunity. And then if it's a good vaccine, but very low vaccination rate, which is probably the outcome for this population, given fake news that's going around and so on, we're not going to end up with herd immunity, which means we may be looking at a third wave or fourth wave and the continuing pandemic. Not to mention what's going to happen with the regular flu that should probably emerge in another 3 months or so. The best scenario, high efficiency and high vaccine rate. I think once it comes down that this is widely available, people will start getting it. Now, zoonotic diseases, and in a sense, we've been talking a lot about zoonotic diseases. These are viruses in particular that are in animal reservoirs and then make a jump to some intermediate animal and then make a jump again. So 75% of the emerging viruses are what are called zoonotic, and we'll talk about some specific examples. 60% of all infectious diseases, this includes bacteria, are zoonotic, jumping from some animal reservoir, which is why epidemiology is so important. I just was with a former student, and I'm trying to convince her to go into public health, because if she had her degree in public health, master's, she would instantly be hired by the CDC. So it gets very complicated figuring all these things out. Where is the reservoir? How does it jump to livestock? How does it get to people? And then the scary thing is once it's in humans, it may just jump from human to human. This is an idea called spillover. There's a couple of very good books. David Quammen, who's a wonderful writer. All these viruses have natural reservoirs, primates, mice, bats. They may have vectors that will get it into semi-domesticated animals, and then eventually they amplify it. And they amplify it because we encounter them more than we encounter monkeys or bats. And some of these viruses will pick up genetic material. So ultimately, smallpox came from camels. Likewise, the last MERS which is the Middle Eastern respiratory syndrome, came from camels. So you never know where the intermediate is. And then these spill over again into humans. And the real fear is that you will have human to human transmission. So many start with bats, not because we're picking on them, because they are volant. They fly from place to place. They're often in very large colonies. But they may get into other things like pigs. We'll talk about Nipah later, and then into people. Flus go from ducks to chickens, and sometimes even to pigs, and back into humans. Here's the MERS, which went from bats to camels on out. And SARS, the most recent SARS, we think the intermediate host was either a civet or a pangolin. So you get these natural viruses in a reservoir. They get transmitted to some domestic animal and spill over into humans. And tracing this all back is really fascinating. Here's the bat reservoir. We don't know in many of these cases. We have had coronaviruses for a long, long time. Lots of different ones that are just sort of like a serious flu. We think many of them came from rats. We know the recent ones came from camels, civets, and actually pangolins. So this is Nathan Wolff, who's a very brilliant, kind of talented Renaissance scientist. He's been looking at how things like Ebola, which start from rats or bats, go into chimps or gorillas and then jump to people. All right. But now with Ebola, there is a human to human transmission. And eventually some of these diseases like dengue can make it to the point where there's human to human transmission. And that is the ultimate in spillover, which is also very, very frightening. If you want to be frightened, there's a very good book, The Viral Storm. All of these are on the initial bibliography. What makes these really problematic is once person acquires this from visiting or traveling, then instantly this can be spread. The reason the main SARS was spread to Canada in less than 10 hours in a flight from Hong Kong, and nearly had a major epidemic from that. It was quite a lethal thing. So we now live in an age when any of these can spread globally through air traffic. Nipah, I mentioned, which is a very strange, variable paramyxovirus. It has a long strain of ribonucleic acid. It was first isolated in Malaysia in 1998 in a village called Nipah. It causes encephalitis and upper respiratory infection, and strongly associated with pigs. The bats urinate in hibernation or resting at night. This goes into the pig troughs, or the bats eat fruit and the fruit will fall on the ground and the pigs eat it, and sometimes even people eat it. And it's spread very quickly across Southeast Asia, jumping to many species, and now there is human to human contact. So this is a very just typical of what we've been talking about. But this also brings up why so many new emerging viruses. And this has to do, simply it all goes back to population. And in the tropical areas, deforestation, trying to make more land to feed people. And this means we're going to contact with some of the wild animals more often, or at least the animals we bring in. We may put in an orchard, but then This will bring in bats, and if we have pigs and horses, then it will pick up the virus that way and then spread, and it may spread to cats or dogs to humans, or horses in many cases, and then on to humans. So deforestation is actually strongly linked to many of these emerging viruses, particularly Ebola, and also some of the others we'll talk about. It spread from the initial cases very quickly up to India. And then from India, if you look, it spread into Vietnam, and then into Africa, and then all throughout Australia. And this is partly because Australia likes to vacation up in Indonesia and Malaysia. So they took it back by just flights. Now, this gets very interesting because the bats, if there's fruit trees around, they'll roost in there. They like to eat the fruit. The fruit may drop on the ground. The pig eats it. If the humans eat the pig, or if the humans just simply have direct contact with the pig, they will get Nipah. And this is an emerging virus. We can track it. By the way, this is not seagulls. These are fruit bats flying out of the roost, which is an amazing spectacle very few people have seen. But getting rid of the fruit bats doesn't help because, one, you can't get rid of all of them, and you need to stop it somewhere else. One of the other effects is the fact many of these clearings are planted into date palms or into palm oil orchards, which is almost unnecessary. Palm oil is highly saturated. It's not a healthy oil, but it is used for many, many substitutes in butter and other kinds of canola oil. You can tap the palm tree and collect sap. So you see here is a young man climbing, tap it, you put a little pottery jar underneath this and collect the sap, but the bats love the sap too. The sap is going to be taken away and fermented into a wonderful palm wine, or distilled into a kind of palm sugar, which is used for lots of things. The bats come and lick this, drink it, and urinate in it. The virus gets into the palm wine. People drink the palm wine. And so it will have a direct human link. The other linkage, of course, is from the bats eating papaya, mangoes in particular. They leave saliva, the fruit falls, the pigs eat it, and this is also transmitted. So when this was first discovered in '98, they actually ended up killing the pigs, trying to stop the transmission. But any other animal, it would probably jump to horses or sheep or goats or whatever they had, likely chickens. So this is where Nipah is now. We don't have fruit bats. This is the range of this particular massive fruit bat. So you can see it's bound to spread following date orchards, but you can see where it has spread all over Australia, especially Eastern Australia, which is semi-tropical, and massive roosts of fruit bats, which are wonderful to watch. Here's Borneo, Sulawesi, Malaysia, Indonesia, and you can see it's already spread up to China and up into northern India. Here is what I was talking about. Here's a fruit bat, by the way. These are when early on when they had to just bludgeon the pigs to death. But here's a simple solution. You tap the palm sap. There's a little tube, just like tapping sugar maple. Well, you just put a little bamboo shield around it and the bats can't get in there. So this has greatly reduced it. It doesn't take high technology in many cases once you figure out the epidemiology of these. So that's simple to do, just put a little cover around the pot. Something you may not have heard of, I was working in New Mexico when this happened. uh, Hantavirus, which is actually all over the world. There's many different hantaviruses. It's interesting because we generally don't call it Hanta. When the CDC went in there and isolated it, they called it Murder Canyon from the place that it was discovered in New Mexico, and the local Indian said, you can't call it that, that will give us a bad reputation. So they called it sin nombre, the virus without a name, but it technically is one of the hantaviruses. It's tied to population of the mice, and particularly a white-footed deer mouse. During certain times of El Niños or other things, the pinon nuts can increase. And when the pinon nuts increase, the deer population increases. When the deer population increases, then they move into local habitations. And they carry the virus and they leave it in their feces. So if you get on top of the case and start sweeping all this up and atomizing the feces, you inhale it and you get very serious respiratory distress. And it gets quickly worse. You'll get fever, chills, and so on. This actually had an outbreak in Yosemite not too long ago. It is sometimes called the Four Corners virus because of the initial outbreak, but now we see it is spread all the way up and down California, mostly in the mountains where there are cabins that are not open till the spring, and the mice are active when people aren't there. I'm dealing with this case myself right now because I was gone for a month and a half and I got mice all over the place. Anyway, there have been a couple cases in San Mateo and Santa Cruz County, and you see down here, but so far nothing in Monterey County. And every once in a while, there will be outbreaks. So beware. This is interesting in its transmission. All over the United States, we call it the Four Corners, and that's because of the population of both pinions and also deer mice, but other mice will carry it, and you can see various forms of Hanta are widespread even in Europe, South America, Africa, and elsewhere, and it is closely tied to the mast or the seeds that they eat, and closely tied to people moving into mountains and also climate. Ebola is the same sort of thing. Bats transmit it to a whole host of things, antelope, shrews, monkeys, gorillas, even in chimps, by urine. And then humans eating these, not so much direct contact, but it can affect them from direct contact, but eating the bush meat, as it is called. And we now know that Ebola very quickly spreads through body fluids, especially during infections. It was first recognized in 1976. It's been studied extensively because it is a massive hemorrhagic fever. It spreads quickly throughout all systems of the body, especially the capillary and vein system, and it just basically melts from inside. And has a very high mortality rate, up to 55%. It was first noticed in the Democratic Republic of Congo and named after a nearby river, the Ebola River. It is very nasty looking. It's a filamentous single-stranded RNA. It has a lipid coat, and it has a number of different serotypes depending on where it is spread. And it burns out very quickly just because CDC and World Health will send medical personnel in to isolate, to track the transmission, and then to basically sanitize the area and handle and isolate all the infected people. If we did this with COVID, there would be no problem, but this is a very costly operation. And part of the problem, this is what people don't like to see. This is how I, one of the first places I take any of my tour groups is to the local market. And these are bats. This is in China. These are monkeys, these are dogs, snakes, these are badgers. And so people eat them. Bats, you can acquire it just from eating. And bats, by the way, people always ask me what bats taste like. And I'll tell you, they taste quite a bit like guinea pigs. But other things, goannas, these are raccoon dogs, monkeys, even owls. These are bats, more bats. And this is the pangolin, which is not only eaten, but the scales are very, very prized because they're supposed to be lucky charms in China. Mice, squirrels. If you have an enormous population, you don't have jobs, you move into the jungle, you eat bush meat, then your chance of encountering an unusual virus is increased. We see how many of these things are transmitted by bats, so they can spread easily: Marburg, Hendra, Ebola, Nipah, rabies. Although the rabies part is highly overblown, it is not that, they're not a huge carrier of rabies. But you see these intermediates, and they are all domesticated animals, or animals that we encounter on a little more common basis. Pigs, of course, horses, camels, but also civets. And these are basically not quite weasels, but they're related. They are kept as ratters, and they're also kept in some cases in Southeast Asia because there's a very, very prized coffee. These civets are kept and they are allowed to go into the coffee plantation at night. They eat the coffee berry, and they eat basically the pulp on the outside of the drupe. And then when they defecate, people go around and pick up the feces and dry it and take the coffee bean out of it. And because of the acids, this is the promotion from the civet stomach, the coffee beans are exquisite. And it costs at least about $50 to $100 a pound for this particular kind of coffee. Now from there, we jump to something that most of us don't like to think about, and this is cancer, and it is strongly associated with viruses of many causes, many types. About 30% of the population will develop cancer in their lifetime, especially if they live long enough. About 50% will die. There are some cures or treatments, and there are many, many that are very common. It is no single disease. I've taught this for a number of years. It's a fascinating subject. There is no single cancer. So trying to cure cancer is an uphill battle. And it is second to heart disease, especially in mortality in women. There are a number of things that can cause cancers. Some people are predisposed with inherited mutations. Sometimes you can get random mutations, often if you're exposed to chemicals, asbestos, vinyl chlorides, PCBs, and so on. Environmental factors. One of the greatest risks is actually in New England because of the bedrock. You need to be careful about going into your basement because radon will leak into the basement. And in New England, many people have radon detectors in their basements because it is a very active carcinogen. Smoking, probably look at this. Tobacco is linked to about 1/3 of all cancers. And viral induced transformations. It's technically wrong to say viruses cause cancer. Viruses prime certain cells for cancer because they transform the cell and get it more likely to tip toward carcinogenesis. So the main causes, by the way, it's just old age. Living causes cancers. If you look at cancer related to age, cancer incidence goes up. You can't see this probably, but it goes up and then it declines. And the reason it declines at old age is because everyone who was going to die from cancer has already died. And if you look at viruses, well, these are actually not quite correct, but it's estimated that if you trace them all back, about 17 to 20% of all cancers can be linked to viruses. We tend to overestimate the things like PCHs and food additives and food dyes, and underestimate diet, sexual activity, alcohol, tobacco, of course. Some cancers are strictly limited to inherited predispositions. People are always looking for particular genes, BRCA for breast cancer, and it is very strongly linked. This one I'll talk about, P53. Many of these may be activated by viral infections. So you may be predisposed, but then have the DNA from the virus insert in this particular gene and activate it as a cancer-causing gene. A cancer-causing gene, well, there's a couple of types. We generally call them oncogenes from oncology, which is the study of cancers. Proto-oncogenes normally promote normal cell growth, but something can occur to cause them to turn into proto-oncogenes. Some of these normally suppress tumors. The body is always out looking for things that aren't right, and white blood cells will also attack tumors. So we have many genes that suppress it, but if you affect that, then it might lead to unregulated cell growth. And that, by the simple definition, a cancer is a cell that has lost its regulation. It is metastatically now dividing repeatedly. All of our genes have repair enzymes. And if these get faulty by an insertion of a virus, then new mutations can be tripped. There are a lot of these. There are growth factors. There are signal transducers. There are a lot of suppressors. We don't want to go into all of these. I will talk about this protein P53, which it's named because it's a protein that weighs 53 Daltons. Normally, a cell will divide, and this process is called mitosis. You may remember that from high school biology. This is very carefully regulated. Then it divides again, and there's usually a set time, interval, and there's usually a number of times. This is called the Hayflick number, how many times a particular cell can divide before suppressor cells and white blood cells come in and kill it. The definition of a mutant or mutagenic tumor is simply a cell line that has lost its regulation. So cancer can develop anywhere. It can develop in here, can develop in here, in here. So it is a stepwise thing. So almost every cancer starts with one mutation, a second mutation that spreads, a third mutation, and so on. Especially colon cancer. We've followed this really, really carefully, and colon cancer is strongly linked to at least 4 mutations, and the genes are pretty well known. This is why we like to look and see if there's any sign of polyps, which are indicative of the second kind of mutation. You may remember this, I hope you do. These are chromosomes. You only see them when the cell is about to divide. The cell then does a little dance with the chromosomes. They go to respective ends, they separate, and they the cell splits. So you see this, here's a new 2 cells from the original 1. This is called cell division, and we have tremendous number of regulators and checks. And the P50 gene is a regulator, particularly right here. It says, should I keep dividing or should I stop? Should I kill this cell? If it gets mutated, then it simply goes into cell division. And there's another check here, and if it passes that check, it will keep dividing over and over and over. And by simple definition, that's tumors. That is what cancer is. There's another regulator called the RB gene, which was named after retinoblastoma, which is an awful inherited cancer of children. It causes tumors in the retina, but everyone has this. It's a normal regulatory gene in cell division. And cancer looks somewhat like a cell that's infected by viruses. This is what a normal tissue culture would look like, but if a virus gets in here, or if it becomes metastatic, you get these syncytial clumps or tumors. So viruses can induce this. Which viruses? Well, there are some that are very strongly associated with certain kinds of cancer. Epstein-Barr, which is a DNA, often trips later in life a lymphoma, which is pretty nasty because it goes after the very cells that are trying to kill it. You may remember the B cell, the one that produces antibodies. This overproduces ineffective B cells. You can also get another kind of lymphoma from Epstein-Barr. Hepatitis B, liver cancer. Carcinoma refers to the type of tissue. Hepatitis C, another kind of liver cancer. Even herpes, Kaposi's sarcoma. One of the reasons we discovered HIV is when people developed HIV early on, it wasn't obvious what was going on. And then in many of these victims, a very uncommon skin cancer, Kaposi's sarcoma, began to show up in gay men in Los Angeles and New Orleans and elsewhere, and they knew something was immunocompromising them. And it was simple herpes virus that nearly everyone carries, but turned into a skin cancer. Papilloma virus, which we really know is strongly associated with cervical cancer and other reproductive cancers, and we now have a vaccine against it. And it just boggles my mind how parents will not let their children get this. Both men, boys and girls need to get this. And then human T cell leukemia, which will bring on a certain kind of adult leukemia. There are at least 40 known oncogenic retroviruses that affect animals. So we're not the only ones. There are, for instance, I remember seeing feline leukemia reports from Carmel, and the Carmel 93921 zip code, and it was rampant. And cats do not live long there simply because they're older population of people. They have lots of cats. They do not get their cats vaccinated and so on. So a lot of AIDS-like viruses, murine, mouse, this is an avian one, cats, chickens, turkeys, all sorts of things that people have to look out for. We think we're the only ones that have to suffer through this, but there's some really serious ones. So this is how HPV, human papillomavirus, works. It focuses at a couple of different places on the P53 or the RB, the retinoblastoma regulator. So the cell simply progresses on to become functionally immortal. And there is, if you're interested in this, there's another wonderful book and actually made into a movie called about Gila lac, the first time anyone was able to grow a human tumor, and everybody would try. It was a young Black woman in Baltimore who had an ovarian cancer, and her tumor grew and grew and grew, and so it was divided up and spread all over the world as a cancer model, and eventually even went to the moon. It's estimated that there are more Gila cells named after her on the planet now than were in her life originally. And of course, the family was never told about this, and the family never collected any kind of money from this, but an interesting story. Now we go to one of the greatest killers in human history, and that is the influenza virus. And we are knocking on the door again for flu season, and we wonder what will it be like. Well, at least everyone's cognizant of the value of masks and social distancing. Flu is a little different. It survives much longer on dry surfaces and other places. It's a very complicated virus because it has RNA in different little segments, and the number of segments is actually indicative of the type of strain. It has two different receptors. One is a neuroamidinase, which is how it docks, and then there's also a hemagglutinin, which is also important for docking, and then two channels where ions go back and forth into this. So it's a pretty simple, it's enveloped. There's the nuclear capsule, and this is the lipid outer layer. We have studied this for a long time, but it is incredibly variable because it has these 8 segments. It is what we call an orthomyxovirus. Ortho means it's regular, myxo means that it likes to go after mucus-coated surfaces, which is why we tend to get flus in the nose and the throat and the lungs, because those are mucus-coated. It's segmented, the RNA, single-stranded, broken up, and it doesn't have a proofreader, and that means every time it divides, there'll be a mutation for every 10,000 base pairs, so that this thing is a mutational factory. And that makes it very difficult because it will mutate in the 2 different surface markers in the 2 different channels. And each of these will cause us to have an immune response. The influenza is extremely resistant to drying and also extreme cold, which is one of the reasons it tends to float around more in the winter. I like this image. You can see the different surface markers. You can see the M channels, and here are the little segments. And this is what we tend to try to go after, this hemagglutinin spike. If we could cut that off or cap it, then we have a way of trying to prevent this from attaching to mucosal cells. This is the surface receptor, and it enables the fusion of the flu virus to the host cell membrane in the nose or the throat or the lungs. There are 3 main strains, and the influenza A is a very common pandemic causing one, and it often affects young people selectively. It has 8 gene segments. Influenza B is not that severe. Almost never see pandemics or sometimes epidemics. Also has 8 segments. And then influenza C, very rarely in humans, or if it is, it's very mild in children, and it doesn't show seasonality. The other two, A and B, do. We can get more detailed when we look at A. It has 18 possible, and sometimes more, hemagglutinin markers. Then if you combine it with the neuroamidinase, it has up to 11 different markers. And then it also has a number of different M2 markers and so on. So when you do the kind of computation, you can get lots and lots of variation. And the fact that these mutate very rapidly will cause these to change and sometimes become absolutely novel. So we're trying to figure out how to make a vaccine when we don't even know what the markers will be. Add to that, that influenza A can go through humans, pigs, birds, horses, seals, and B can go through humans, seals, ferrets, and so on. And every time it goes through, it can pick up new genes. We name them according to the type. So virus A, Hong Kong A, you may remember that in 1997. The place it was first isolated, the strain number, the year it was isolated, and then looking at the type. This is hemagglutinin 3, neuroamidinase 2, or H3N2. And here are the 8 segments, and these are the genes they code for. These are polymerase to allow copies of the RNA. Now, I apologize for this. It looks pretty complicated and is, but think about this. Here's the influenza A with actually there's 18 of these and there are 11 of the N variations, but just take H1 can go from this way, from ducks to pigs to people, or this way, ducks or chickens to pigs to people. And every time they do that, they will likely pick up some of the genes inside the duck and then mix it with what's inside the pig and then backwards. And it can go this way too. So we know H1N1 has gone through at least 1, 2, and maybe more. Sometimes it goes through Asian swine and European swine. H3 can go from horses to ducks and so on. H7 can go from horses to ducks to seals. So this gets very complicated, and even at times can go through mink on mink farms, whales, and even dogs. If you take this in consideration with the 2 other forms, the B virus and the C virus, the C flu, and the D flu, which is one I didn't talk about, these sometimes intersect. So every season, these things go through 1 or 2 and then come back this way, that way, and come back to people. So they mutate almost within the hour. And I love this, even they've been tracked through ostriches and guinea pigs and weasels and dogs and so on. Sometimes there's very simple direct transmission. That is, you have duck farms, or even if the duck is flying somewhere and lands on your pond or just flies over and defecates, it can be transmitted simply that way. Sometimes they adapt and go from pig to pig to pig, even Asian to Eurasian, off to people. And then sometimes they reassort because it can come back, and that the human virus goes into the pigs. So if we look at this, take a little bit of time to look at this, here are the virus genes inside a duck. And then it goes inside another wild duck. This may be a domesticated duck. And then it goes into poultry. And if you follow these genes, each one of the 8 segments, they can reassort so that the virus that actually ends up coming out of these birds is a mix. Those are domestic poultry. There's one from a duck and so on. So you get a completely new form. And this happens all the time. And you think of where these come from. This is how people live in much of the Asian world and even in Africa. You live, you don't want snakes and other things, plus you want animals to have shelter, your dogs, your goats, chickens. Often your bathroom is somewhere back here because you just defecate right underneath the house, and the pig is very handy at eating that. It's not discriminating. Then this somehow contacts the chickens, or this washes during heavy rain into the local duck pond. So the virus goes from a human to a pig, down into a chicken, and washes into a duck pond. So it's now in the duck. If there's a wild duck, it might fly away. So every time I look at these things, here's pigs again underneath this stilted house. It's just primed for making new viruses, new flus. And how do you track it back to any of these? How do you change people's lifestyles? You can't, because these are poor people. This is the way they live. There are 2 ways you can change the virus. You can have a very small mutation, sometimes a single base pair. And this is sufficient often if it affects the H and the N spikes. So you get a tiny little mutation, and any immunity you had from previous viruses were against this form of the H or N. Now you have no memory of this, and you have to go through the whole T cell recognition, dendritic cell eating, macrophage, presentation to the B cells making antibodies, and that takes time. And so this is very common, this simple, small mutation, and these are often will cause serious epidemics, mostly with the A and B viruses. Now you can get a complete shift. You can really change things. Mostly this occurs in the A subtypes, and it happens suddenly and explosively, so that the new subtype is completely novel. It's a mix of 3 or 4. It may be part chicken, part pig, part Eurasian pig, and what comes is so novel, no one's going to respond to it quickly. So this is a pandemic causing antigenic shift. Remember what the antigen means. These little H markers and so on are the antigens that the B cells are trying to make antibodies against. It may have seen this one in the past, but it has never seen this one. And so when we look at going back, and we have records and we've dug up bodies so that we know way back in the Russian flu in 1889, we know what that was, H2N1. And then when we come up to 1918, we know this, and it's very likely the reason this was so deadly is that the H3N8 had been around for almost 20 years, and that's what everybody built up immunity to, and then suddenly there was a new form. So this lasted a long time, then suddenly this new form, and nobody had antibodies against it. And so this was one of the biggest pandemics in human history. Now, it looks like this came out again. It did, but it's not the same. Plus, people have some inherited antibodies against it now. There was a near miss with this one in 2009. And we will talk about some of these, but notice these all are kind of understandable when you realize what we're talking about. So any flu, this was a 2009 flu, which was quite deadly. It was reassorted so that there was some human DNA or RNA in it. There was chicken, there was American pig, and Eurasian swine. And think of all the permutations of mixing these and what kind of antigenic markers you're going to have and how these are going to work in the body. We know enough about the different pandemics to know how they got reassorted. So here is this reassortment from 2009, ostensibly in H1N1, but look at the genetic variability in here that's going to cause us to be infected and produce lots and lots of new ones. You can get triple assortment, which is what I've been talking about. So this 2009 really is a triple assortment from different combinations of ducks and chickens to different pigs. And so this is what we had to deal with. The flu is highly infectious. It can transmit through sneezes and droplets, but it's also very tough and it will survive on surfaces. It can survive for 2 days off of the host, multiple strains. It can be transmitted by contact with pigs or waterfowl, shoes, clothing. Imagine just working in the farm, coming in, feed cages, can survive a month in bird feces and probably pig feces. So, and this is a very typical view in Southeast Asia, walking the ducks and the geese back and forth between the house and some sort of pond. And then here's the pig and the chickens, everybody living together. So flus are incredible. And flus, if you can walk around and talk that you have a flu, you don't have a flu. Colds are extremely common, more than 100 different viral strains. They tend to be gradual. They tend to be upper respiratory, that is airway, the nose and the throat. Some headache, general aches, a little bit of a cough, sometimes a mild sore throat and so on, but you're never fatigued or exhausted. Whereas flu, you will be down. You will have high fever, you'll have severe body ache, you'll have severe cough, and this may last for 2 or 3 weeks. So flus are very, very different. And this is a common rhinovirus, mean one of the adenoviruses that affect colds. Now, this is an interesting thing. We don't know how many people actually died in America or in the world. We think at least, this is probably wrong. This is the Spanish flu, Asian, the type A, N1, H1, probably more. This is a low number, probably 1 to 2 million. This worldwide figure is probably off. It's probably off by 20%. That is, we had no figures, nobody, no World Health Organization. But look, 1957, and some of you may remember, 2 million people died. Hong Kong flu, 1 million people died. The 2009 flu, 250,000. So, and interesting, this is one of the strangest things. In the 1918-19 flu, it was young, healthy adults. The Asian flu, elderly, I hate to say over 65 is elderly, but anyway, Hong Kong flu was also this way. And this is what COVID is doing right now. And we may be seeing a shift to this because college campuses, nobody seems to mind partying without a mask or anything else. And so now these people are getting sick. They may not have the same mortality rate. How did the 2001 start? It actually started in the United States, went up north, came to England, came to Asia and into the Pacific, and had major outbreaks across the world in less than March, April, May. In 3 months, it spread to 53 countries. So that's the potential, and you can see what aircraft can do. Now we come to the mother of all epidemics, the 1918-1919 flu. When I was a kid, I worked on playgrounds with supervised kids, and I often wrote things down about what the kids would do. I had a little bird and its name was Enza. I opened up the window and influenza. Kids were still saying that. No one really knows where it started. Maybe in Kansas, maybe in Europe, maybe in Australia. It came in in Southeast Asia, maybe in 3 different waves. And we see this, and this is what people are talking about. This is probably what we're going to see October and November. This is exactly what we're going to see, a really big second wave, and then probably a third wave. Much more fatal. And it killed, this is age, it killed young people. And none of this should seem unusual. Schools, theaters closed, churches, masks required, sometimes very innovative masks, and I don't know what that was. And some cities closed down. Here was St. Louis, closed down almost immediately. Churches, public, and look at the mortality rate. Interesting. Do we ever learn our lessons? Not really. Gargle daily, wear your masks. Spit, you could be fined for spitting. Same thing happened. Football teams wore masks, baseball teams, you could be arrested for not wearing masks. There were anti-mask leagues. Nothing has changed, except now it is viral, excuse the pun. And we're going to have to live with this sort of thing for the rest of, for a long time. And it's mostly due to social media and conspiracy theories and misinformation. There are a lot of books. It's a fascinating study. There's a great number, and I sent this out. I forgot to send this out, but there's some wonderful fiction which will show you things were bad in the past and we recovered. We do have a vaccine against it. The first pandemic, then we made a vaccine, very simple vaccine in the 1930s. It's a complicated process. We have to look at the strains that are coming in all over the world, then make a decision of what to go ahead with, and then produce it and distribute it. So this happens every year. In the past, we used to do 3 major strains, but since the 1980s, there are 4 strains that tend to spread. So you're dealing with 4 different strains that are often, well, probably more, because we don't know which one will make it to the United States. For kids, it's not a live vaccine. It is for older, and then for elderly, it's not. And it's also a trivalent, not a quadrivalent for older people. We do have flu drugs, and you know, you've heard of Tamiflu, which is not without its side effects. And these work by blocking the H receptor or the N receptor, but already we have some viruses that are flu viruses that are resistant. Now, more to worry about, many new viruses that are emerging. Some I've talked about, Nipah, SARS, MERS, dengue, chikungunya, Lassa fever, which I didn't talk about, another hemorrhagic fever. And not only just viruses, there are bacteria that are getting resistant to antibiotics that are emerging. These are the ones we are mostly concerned about because we know about them. But it's the one we don't know about that's probably going to take us by surprise. A couple of cautionary tales. 2003, a pet dealer imported giant Gambian mammalated rats. He kept them along with his prairie dogs. Then he shipped these animals up to a number of states in the Midwest, and the Gambian rats transmitted monkeypox, which they picked up from monkeys, to the prairie dogs, and they spread monkeypox to pet owners. There were 37 confirmed cases and 53 suspected cases, 37 confirmed, and it is a pox. It's not a fun lesion to get, and fever and so on. This has subsequently happened in the United Kingdom. Close call. CDC and FDA said you can't bring any of these animals in anymore for the pet trade, no matter how cute they are. And if you travel in northern Florida, there are rhesus monkeys or rhesus macaques that have been there for quite some time. They carry herpes B. We don't know quite how they got it. They were probably introduced in the 30s in pet parks and so on, but they are carrying a very nasty herpes, and sometimes they can interact with people. Let us not forget the Reston virus, one of the 6 Ebola types. It was sent in with crab macaques in 1989 to Hazleton Laboratories. These monkeys were also infected with simian hemorrhagic fever. And so a couple of people who worked here came down, but it was the hemorrhagic fever that was probably the cause, because the Reston virus doesn't affect people. But if you really want to scare yourself, you can read about the hot zone, which is a good account of what happened to this place. So what's going on? Well, let's look at population. We can't get away from it. And where is this population going? They're digging into the rainforest. They're building anything that they can grow for food. They eat anything they encounter. They're making this absolutely a melting pot for future viruses to emerge. So when all is said and done, viruses are probably the oldest life entity on the planet, going back 3.5 billion years. They occupy every place. They are probably the largest genetic diversity of anything on our planet. They have remarkable reproductive rates. They evolved inside everything and helped everything else evolve. And they transferred DNA and RNA between all sorts of animals, and they still do it, to the point that about 50% of our own genetic material is virally derived. So I'm always interested in how we respond to things, both in art. I'm very interested in what kind of literature will come out of this. Not much has changed. It is trying, it is tiring, but so far our world is temporarily closed until this all gets resolved.